Thank you very much to the organizers for the great opportunity to present you one of our studies. So I'm going to switch on gears and I'm going to talk about the therapeutical potential of spider peptides and more specifically on the anti-cancer properties of gomecin peptides.
Are you sharing your screen already? Because we just see your PowerPoint. I think we're having the same problem we had earlier.
Oh, let me see. Okay. How about now, Anna?
Oh, great. That works. That works.
Perfect. Thank you. So this is one of the gomecin peptides come from the Australian funnel web spider that Glenn very nicely introduced yesterday. And he highlighted the complexity of the venom and the therapeutic potential, especially targeting ASIC channels. But I'm interested in their anti-cancer potential. So, and specifically for their properties in targeting melanoma cells. Just a little bit of background about melanoma. It's the main skin cancer that causes fatalities worldwide. It affects both genders, and even though it's more prominent in males, but that's probably more due to lifestyle and work related reasons. So the main melanoma mutation is the BRAF that is found in V600E position. And even though at early stages is treatable by surgical removal, a big problem is that when it's advanced or it's metastasized, then it's lethal. And that's where all the problems occur. So the BRAF pathway, when it's activated during cancer in melanoma, then it causes uncontrollable cell proliferation. So there are different inhibitors that they're trying to stop, to inhibit this pathway, its activation at different steps, as you can see. So the current therapies are started with monotherapy, which were BRAF inhibitors or MEK inhibitors. And then they just moved on that they found more effective were the targeted approaches when they combined the BRAF and the MEK inhibitors, and they prolonged the lifespan of the patients. Even better results were achieved with immunotherapy. And we've got the CTLA-4 blockers, for example, or anti-PD-1. And combining immunotherapy as well as triple therapies is something that is currently investigated a lot with quite promising results. However, there are some challenges. A lot of patients are unresponsive. In immunotherapy, I think it's like 30% of patients, and there's a lot of adverse effects. So many times the clinicians have to stop the therapy because the patients cannot continue due to the toxicity levels. Obviously, due to cancer, is many times they will have the issue of drug resistance. And you block one pathway, another pathway is activated in order that the cells to keep on, the cancer cells to keep on proliferating. And eventually we see that the cancer patient is relapsed. So there is still room for new approaches, for new melanoma therapies. And that's where it comes our interest in the gomecin peptides. So the first one is very well known and very well studied. It comes from the hemocytes of the South American spider. It's 18 amino acids long. You can see just in the maquette below, the structure is beta-herpin, is very stable. It has 4 cysteines, and it's a pore-forming peptide. And it has been investigated for many different properties, including anti-cancer. The other gomecin peptides that it was found from the transcriptome of the Australian funnel web spider from the Venom lab by Sandy Pineda. It's very similar, as you can see in regards to the sequence. There are only 2 residues different. And we had investigated and we have confirmed that it has both antibacterial and anti-cancer properties. We don't know if it has more. So as you can see, as I said, it's very well studied peptide for their anti-cancer also properties. There's a lot of work how you can modify it to be more stable, more potent to enter the cells, and of course for their anti-cancer activities that they've been reported. So previously we saw that both of these peptides, so the agom is the Brazilian peptide, spider peptide, while the higom is the Australian peptide. And that's where I'm going to refer to them from now on. So we observe that there is a more specificity towards the melanoma cells, the MM96 cells, the BRAF mutated, in comparison to the healthy human fibroblasts. And we confirm that more or less, these two peptides have a similar pattern in a range of BRAF mutated melanoma cells. So we set some further light in regards to the affected signaling pathways when they enter the cells. And we saw that it affects a lot the Hippo pathway, which is responsible for the organ controls, proliferation of the cells, inhibited the MAP kinases, and it activates contradictory the AKT-mTOR pathways. And I say contradictory because these pathways, the signaling pathways, are responsible for cell proliferation. So we hypothesize that this is probably happening as a mechanism that these melanoma cells employed in order to try to avoid the cell death that we saw later on that it actually occurs, which is a very common technique, I would say, for cancerous cells. What we are doing now, and that's very preliminary data, so maybe they change later on, we're interested to see how the metabolic profiling of the melanoma cells is altered due to the gomecin treatments. And Nayara very well introduced the Seahorse technology, which allows us to see changes that they occur in relation to different glycolytic parameters. And I just have listed before some of them that we study in our group, but also in regards to mitochondrial changes, to the mitochondrial respiration linked to the oxidative phosphorylation changes. What we did observe when what you can see in this slide is that the AGOM, the Brazilian peptide, seems to affect both and the glycolytic parameters, but also the mitochondria. However, when we compare it with the HIGOM and some other gomecin peptides that I'm just going to introduce a little bit later on, we saw that this one doesn't seem to cause any metabolic changes, which is very interesting. So first we need to confirm that this is the case by doing some more replicates. But if that's the case, I think it's very interesting to see how, and it will highlight how 2 amino acids changes can have such a tremendous impact, on the biological activity of the peptides. And this is the work that Javier is doing. We had obviously been able previously to confirm the therapeutic potential of the HIGOM in nude xenograft mice. So when we induce the cancerous cells by subcutaneous injection of the cells in the mice, and then we treat them for a short period of time, a little bit over a week, we observed that the tumor progression was much slower in the treated mice in relation to the vehicle controls. So, and this was a further also verification of the therapeutic in vivo potential was made in Laura's lab in San Diego de Compostela, where Pablo were injected the melanoma cells that he had previously labeled with fluorescent staining. He inject them in the circulation of the zebrafish embryo, which is a very good model also for studying toxicity or also the anti-cancer potential of a toxin or a venom. And we observed that, or they observed that the melanoma cells were proliferating at a lower rate when they were treated with both of the gomecin peptides in comparison again to the vehicle treated. So we were very excited to have these two independent animal model confirmation on the anti-cancer, or more specifically on the anti-melanoma potential of our gomecin peptides. As I mentioned before, we have also tested some different gomecin peptides because we observed, as you can see, that there only, as I said, 2 amino acids difference. So with Andreas Brast from Paul Alwett lab at UQ, he synthesized this panel of gomecin peptides, based on the differences. Mainly you see the first, 6 on the differences of the 2 amino acids that distinguish the Australian from the Brazilian peptide. The only outlier is the 3rd from the top, which is actually coming from a Selenolupus spider, Australian spider, is a big spider. And it has naturally, this mutation. So it's a naturally found gomecin analog, again from the venom gland of the spider. And again, this was work that Sandy did in Glenn Skin's back then lab. But with Evelyn, she was interested also to see if there are changes, if you change the hydrophobicity or the electrostatic potential of this peptide, but just doing some very subtle alanine substitution, as you can see from the 3 last peptides, if you can also have an impact on their cytotoxicity. So we did synthesize all these peptides, and then we just tested them at high concentrations at both melanoma again and the fibroblast cells. And we observed that most of the peptides, the gomecin peptides, seems to be totally selective and potent in targeting the melanoma cells, but much lesser to none effect on the fibroblast. So that was very interesting and very welcoming therapeutic aspect of property that they had. The ones that didn't have any impact were the ones that we altered the alanine. We had the alanine substitutions. So then what Evelyn did, and I had published all this work, so did some molecular stimulations, trying to understand if there is any link between, for example, the structural flexibility of the peptides, which she observed that by doing MD simulations, that the gomecin, the agom, was more flexible than it was shown by NMR structure. And then she compared it with the other analogs, but we couldn't find any direct or obvious link between the cytotoxicity and structural, the conformational flexibility of the analogs. She also looked at the electrostatic potential, and again, the picture was not very obvious or clear, so we didn't find that there was any direct link. So we thought with Evelyn that maybe we do know that the gomecin peptides form forms, or at least the gomecin, the agom, and it interacts with the lipids in order to enter the cells. So she has set up these assays with artificial membranes, which she used this fluorescent probe, the RH421, and where she can indirectly measure the binding of the peptide. In regards to hypothetical, as you can see below, phospholipids and lipids ratios. So we did a few selection of a few potential lipids that might be present in the cancerous cells. And what she observed is that the POPs cholesterol seems to increase the binding of the peptide in the members. And this is a highlighted that is a well binding, shows that a well binding. So she also has another indirect measurement of measuring using this method, the leakage or the disruption of the artificial membranes. And then she observed that, and that correlates well, which I'm going to talk a little bit later on about our interest in cholesterol and the role that cholesterol might play in the cytotoxicity of the gomecin peptides. So she observed that if there is no cholesterol, there is a high, she observed that the peptide disrupts a lot the membrane. However, when you increase the cholesterols, then it seems that it reduces, it inhibits this disrupting effect. And that was very interesting. She recapitulated these assays using the HIGO peptide, and similarly showed that the POPs cholesterol, they cause the maximum binding of this peptide in the membranes, and also that the POPs seems to have an effect without the cholesterol. But they did observe similarly that the cholesterol, increases of cholesterol concentration does reduces, the disrupting effect of the peptide in the membranes. So back to the interesting cholesterol, Javier looked at it and he was able to observe that, that these peptides have these domains, the crack domains, that they indicate that they're binding with cholesterol. So both of them, the AGOM and the HIGOM, they have it, and some of the other gomessin analogs, some of them don't have it. So we are trying now to understand this correlation better and experimentally validate it. So Javier, in order to try to recapitulate, if possible, you know, the result, the data that they were generated by Evelyn in her artificial membranes with a more biological context, he set up this viability. He did these viability assays where basically he kept the ratio that that Evelyn had that shows that they cause the maximum binding effect of the gomessin peptides, at least from the 2 gomessin peptides. And he observed that while you increase the concentrations of this ratio, then you lose the cytotoxicity of the peptide. And I just have to say, when I say here control, it's just the peptide alone. It's not any PBS or anything like that. It's the AGOM by itself, in this case, the HIGOM and so on. And this trend was observed in HIGOM, but it wasn't statistically significant. But we did observe that, you know, the other peptides that they were also inactive, we saw that they weren't affected by these increases in concentration. Another peptide that is interesting because it looks like it has a different mechanism probably of action is the one that is come from the selenolipids that I mentioned. Because this one, even though it's not cytotoxic, you know, at a lower concentration, however, when you increase this ratio, the concentration of this ratio, it looks like it increases cytotoxicity. So just to verify and understand more on the role of cholesterol in the cytotoxicity, we did the same pretty much experimental design as Evelyn did. We used, of course, 300 micromolar concentration instead of, I think Evelyn uses much more in millimolar ratio, so to make it more biological relevant. And he observed that when you add cholesterol, indeed, the cytotoxicity is reduced. And that was also occurring with demosterol, which is the precursor of cholesterol. And this was the same observed with both of the gomessin peptides, but also a totally contradictory result with the SP-GOM, which also SP-GOM doesn't have the crack domain. So it just looks like it's acting in a different way. And we use also LPDS, which is a lipid deficient serum, which basically extracts all the lipids that they are in cholesterol in the membrane of the cells, in this case of the melanoma cells. And we saw that when you extract it, you know, you have it's cytotoxic, but when you start adding cholesterol or its precursor, again, it buffers the cytotoxicity. So what we did then, Isabella from my group, she tested the total cholesterol level that is in melanoma and in comparison to fibroblasts. And very interestingly, she showed that the melanoma cells have a very low cholesterol content, total cholesterol content, in regards to the fibroblasts that they have a high cholesterol content. So, and just like that, to put it into perspective, when we added, you know, the gomessin peptides, we saw that it doesn't have any effect, the gomessins, it doesn't have any effect, they don't have any effect on the total cholesterol levels. But interestingly, the fibroblast seems to reduce, the gomessins in the fibroblast seems to reduce the total cholesterol levels. So now we're following that up to see if, for example, if that's the reason for that, cholesterol, if that's the reason why gomessins are not active in the healthy fibroblasts. So if we increase a really high concentration of fibroblasts, high concentration, of gomessins, then maybe we can see that it reduces the cholesterol content, and that's why it's cytotoxic. So that's the follow-up experiments that we're doing. We also use a drug, is beta MCD, the shortcut, I can't even pronounce the proper name. And what it does, it disrupts the lipid rafts that they usually play a role in the mediation of the lipids with the peptide. And we saw that when you disrupt all these lipids that they are in the cell membrane and the cholesterol is free and sequesters in the membrane, then we see that the cytotoxicity of the gomesins is vastly enhanced. And that is really, really interesting in understanding the role of the lipids and cholesterol and how the interactions they occur in order to gomesin to be cytotoxic. So this is one part that we're really interested in. Another approach that we're doing in order to understand the cytotoxicity of the gomesins is CRISPR knockout. So this was work that it was done in Craig Neely's lab at the University of Sydney, where Sergey from his lab, he actually did a whole genome knockout using the CRISPR-Cas9 technology, using one of the high gomesin. And he was able with his methods, and after that, using ingenuated pathway analysis, the IPA analysis, to determine or to identify 5 cytotoxic genes using very strict conditions. And we also included in this one more that it wasn't really so potent with IPA analysis, but it is interesting for us because it's related with the cholesterol. Now, I don't have yet any data, but I can say that Javier and Isa, they have done a heaps lot of work with shRNAs individually knocking down these genes, each one of these genes, and then testing the gomesin. And we do identify, I mean, we have some candidates, we just need to confirm. 3 seems to be the more prominent and to be in both of the gomesin peptides responsible for the cytotoxicity. So then we will be able to have a very good idea which are the genes and the pathways involved in the cytotoxicity and the role of gomesins, the role of their role in cytotoxicity. So some conclusions is that the agom does not bind to or disrupt to the POPs lipids, and it does require the POPs to bind and disrupt the membranes. Cholesterol that does not necessarily reduce the binding, but it reduces the disruption even in the presence of the POPs. While the high GOM seems to have a weaker binding in comparison to the agom to POPs, but it shows very strong binding and disruption in the membrane in the presence of POPs. And cholesterol seems to slightly increase this binding to the membranes, but reduces the disruption even in the presence of POPs. So cholesterol also seems to blunt the gomesin cytotoxicity in the melanoma cells. And we think that disruption of the lipid troughs, as we saw with the Vita MCD, seems to increase the peptide's cytotoxicity. And this is like the hypothetical, probably mechanism of action that Javier has about how they're acting these peptides. And now what we're doing, the follow-up steps. So we have work in progress, lipidomics, which they will able to give us the profile of the lipid profile that exists in both in melanoma and in the healthy cells, with and without all the different gomesins, the different gomesin analogs, the active and the non-active, so we can link it directly to which are the key lipid players related to the cytotoxicity of the gomesins. Also, we will have soon very data in regards to the target of the genes that are responsible for the cytotoxicity of the gomesin, of the high GOM from the CRISPR knockout from Sergey's work, and how they mediate the cytotoxicity. And another work we're doing now, we're doing a lot of microscopy. Well, Obedani Kariotti, who is now a new group leader at IMB at UQ, he's actually he has labeled argomesin, and he's going to see how it enters these melanoma cells, and maybe we can do some more work in the mechanism of entrance, how it enters, which is the pathway it follows in both melanoma and fibroblasts. And just briefly, this is the work that there are a lot of collaborators that they synthesize this peptide, or that they have an active role in this project. And of course, the people from my lab. And you can see Greg, I mentioned, and Laura is a very good collaborator. She's doing all the zebrafish models, animal models. And of course, every one of you for the attention. Thank you. Let's see if I can stop it.
Thank you so much. There's a question in the room already.
Hello, Maria. This is Esperanza. Super nice talk. It's really good to see toxin-lipid interactions back on the stage. I have a question about the composition of the membranes, because something that I missed on the mixture was the sphingomyelin, especially if you are talking about lipid rafts. What do you think? Are you planning to include it in the future?
Yes, actually, Esperanza, I was hoping you're going to ask me. So yes, I do know that sphingomyelin has probably a significant role, but we started with these ones first, just as a starting point, because it can be like all kind of different lipids, you know, potentially present in this melanoma cell line. So based also on the lipidomics, that's why it will be very important to see the lipidomics profile in the melanoma cells. I think we will have to do some work with artificial membranes, or probably Evelyn has to do some work using the sphingolipids in the cells.
Just a small note on that, something that we saw in actinoporins and the Ikonomopoulou toxins is that they really like the boundaries between the lipid rafts and the more solid parts of the membrane. So it can be that a curvature have an effect on the toxicity. That's why I was asking specifically about the sphingomyelin. So, but if you are already on the lipidomics, you already have it on the track. Thank you again, Maria.
No, thanks, Esperanza. Thanks.
Are there any other questions in the room? I saw a question pop up online in the chat, but I don't know if that person wants to say it out loud or if you want to read it for them.
Okay, I can read it. It's from Nayara. So she's asking me if we use planar lipid membranes as artificial membranes. They just use, artificial totally membranes. So haven't you noticed the curative sensitivity property of this? I mean, I'm not a biophysics and I'm not an expert in the method. So probably Evelyn will be able to answer that, but this was more of an totally cancer membrane potential composition. So I'm not sure I can answer about the curvature sensitivity property of this one, Nayara. I have to ask Evelyn. You know, I don't know if it can be additional information coming out instead of just you know, what we observe in regards to the binding.
Okay. No other questions in the room? And no other questions online? Okay, then thank you so much.
Thank you.